we had was the type of, the pod that the whales came from. But we will have that, we'll gather that data as much as possible when we do the big sample. It could be, yep. Yeah, and back.
I was at a clinical years ago and I heard that speak, and it was my understanding that there was already something going on then with the Endangered. And I felt like she was talking about it, or that someone else was talking about it, regarding examples. And so.
Oh, I'm having a hard time hearing you.
Sorry, I heard Catherine speak a few years ago. It was my understanding that there had already been testing on fecal samples with regard to PCDs, PDEs, stuff like that. So I'm confused about why there's two now when I feel like I've heard data about feces and this is organic fluids before. But I don't. Is it a new way of testing them? Are they? I don't know. I'm trying to put my brain together.
Yeah. Did everybody catch that question?
No.
No. The question is, in seeing Catherine speak a few years ago on the study, she had mentioned some toxin work that was being done on the whales. I don't know if she specifically said if it was on the fecal samples or not. There has been a lot of toxin work done on the blubber biopsy samples, and that's where a lot of the foundation of our work is able to stem from. Really important work. What our study can do that's a little bit different is get more samples. The biopsy samples were based on smaller numbers simply because they're harder to get, a little more invasive, whereas we are getting hundreds of samples a year that we'll be able to look at that on. I don't know of studies that have looked at toxins in killer whale poop, but there have been studies that have looked at toxins in scat of other species like sea otters, some other animals within the area. So that maybe is what she was referring to. But this is the first kind of looking at these scat samples with the full toxin work. Yeah?
I guess a couple questions then. Do you know the ages from that sample size of 2?
We don't know anything.
No, okay, just that.
The genotyping was able to narrow it down, or maybe it was from, I don't know if the genotyping, when the samples were collected, it was known they were following those whales.
Okay.
So that's all we know on those.
And the other question then, you know, your hypothesis is that you're going to see reproductive failures as toxin levels go up.
Yeah.
And this is great. I mean, obviously your methodology is non-invasive and it's longitudinal, it's awesome. But then you have to kind of compare that to anecdotal or observational data as to who's had what offspring in the family line, right? Because is there any measurement you can take that shows a historical birth rate for an animal?
There is. The question is talking about my hopeful data slide, my expected outcome slide. And if it's, I think your question is, it's not going to be quite that simple. And can we pull in historical data on the samples we're collecting themselves? Like, where was that whale a firstborn? Is that maybe your question? Some past family grouping of that. And of course we will. And there's been some really great modeling that's already been done. There's some great publications out there. So we'll build upon what they've done and add in our toxin measures and some other measures. But you can add in family groups and firstborn and pod and other measures, past measures that have been known about that family group, and use it to build on the big story. Chances are that if the driver of the model are simpler things, it may not even be necessary. But I know that age, gender, pod are all really important factors.
I guess I'm just wondering, how are you going to measure reproductive success?
Okay, how am I going to measure reproductive success? Using historical census data, which the Center for Whale Research has collected, it gives us an idea of what an expected birth rate for a reproductive age female. And so we can take how many calves we can expect that whale to have had and compare to how many she's actually had, and create kind of a ratio, and then start to correlate that expected versus observed to actual toxin level. And as we'll start to see patterns with that. Yeah?
How about DNA samples to get a little more specific about which whales are more contaminated and reproducing more, following their offspring as well?
Looking at actual genetic ID for the whales?
Yeah, can you do that through fecal samples?
Yes. So the question is talking about genetically genotyping the whales using the fecal sample, and that is something that's being done. It's in collaboration with NOAA, the Northwest Fisheries Science Center, and their groups. There's a genetic library that's being developed. About 60% of the whales are in it at this point. And this data, we take swabs of all of our poops and we send it to NOAA, and they run it through their equipment and tell us exactly which whale that poop came from. And it really adds, these collaborations add to what we're able to do with this study, because we can be behind 20 whales a quarter mile away, see a poop, scoop it up, and then through these collaborations, we can find out exactly who left the sample. And then that builds into things like the modeling and understanding age, gender, family relationships, and how that can be used to understand their toxin level and stress levels and all these other things. Yeah.
I'm curious as to whether there's parallel studies being done on toxicity within the food chain and of the salmon tissue samples or blubber samples, or not that we're chasing salmon for their food, but.
Right. The question is if other parts of the food chain are also being tested for toxins, and they are. This is work, I know NOAA is doing some really great work on this, and there's some other groups as well. It's a lot to do all of it for one of us. But in collaboration with other work that's being done, we can relate the toxins that are being studied in these salmon levels and relate it to what we're seeing in the whales. So absolutely. It's all part of the big picture, really. Yeah.
If the stress levels are lower at early spring when the whales are returning to the sailor sea, is that kind of an indicator that their food source is more available during the wintertime when they're not here?
It led to a lot of questions, absolutely. The question was, if with the stress levels being a little bit lower when the whales first came in the spring, what does that tell us about their prey availability in the winter? Is that right?
Yeah.
And this is where some of the questions came up of the importance of the early season Chinook runs, Fraser River Chinook runs, possibly the importance of Columbia River runs, which a few of the scat samples collected by the NOAA group have shown that the scat came from, or show that they had been eating the Columbia River Chinook. And it may be some others, but they're coming in a better nutritional state than I think was expected, especially by Catherine and Sam. So it's raised a lot of questions, opened a lot of doors. It's interesting.
Yeah, this is related to the same question, and it's one of the things I was thinking about in the early part of the presentation. In the early spring, first question is, what is the early spring? You know, when are you doing the data? Because this is month to month is fairly important there between early Skagit fish, very few Nooksack and Fraser early fish, lots and lots of Columbia fish. So my question is the time that it's done, but also, is there a difference between, have you teased out with the data, JPOD, who is here through April and into May, so that timeline is pretty important, where historically Ks and Ls are showing up. We like to think the third week of May, but usually sometimes it's not until the first week of June, when they are out off the coast, out off the Salmon Freeway, coming past Swift Shore and down to the Columbia, where there's a lot of Columbia River Chinooks. The more Columbia River Chinooks, the later it is that we see J's and K's. So what was the date, what you call early spring, and who were the whales? Was it JPOD, or was it K's and L's? Because the graph doesn't indicate, and I think the data would be dramatically different.
Yeah, it's a great question. The question was asking about being more specific and defining early spring by month, by pod, by how the other runs are doing, not just the Fraser River Chinook. And that's a question that has come up definitely. Catherine talked about this a lot in the work that she's done. She didn't have as many early season samples because they're harder to get and getting the team on the water. And so it's something that with this new round of funding that we've made a priority to get out earlier, so we can increase the sample size earlier so that the models can handle teasing out things by pod and by individual and by other runs. At this point, the data couldn't handle modeling too much, but we have trends. So right now, we have enough to know we really should look further, which is a great start. But it's something that's become a priority with the study.
One more.
Yeah, there's been a question over here for a while.
What's the average of orcas that die each year from toxins?
What's the average each year of orcas that die each year from toxins? That's a great question, and I would love to better understand that. We don't know. Hopefully this data will, doing more work on this toxin values will help us better understand it, or tease out how important it is, or how much it really is affecting vital status. We're still working it out though.
And why aren't you doing K-pop too?
Oh, on the data? That's a good question. Why don't we have K5? We did 2 samples to validate our methods, to make sure the system's going to work. And we happen to do a J and a K, or a J and an L. But now that we're doing all of the samples, we'll make sure we represent everybody. Yeah.